In this deep dive, we are zooming in on the most dynamic part of the cell membrane. Hello and welcome back to BioInsights. Across this session, we will decode the fluid mosaic model, master the specific lipid to protein ratio, and categorize the membrane proteins by ease of extraction. Let's get into the details. Every living cell is enclosed by a thin dynamic living boundary that is selectively permeable and flexible. It works as a barrier between the inner and the outer environment of the cell. Now, why I emphasized on the word the living cell? Because the specific unique traits of the cell membrane are often compromised in a dead cell. For example, a living cell membrane is selectively permeable, but a dead cell membrane loses that ability and it becomes leaky. Now, the term cell membrane was coined by Nageli and Kramer. The cell membrane, also known as plasma lemma, that was coined by J.Q. Plowey, and it is known as plasma membrane as well. Now, before we directly dive into the fluid mosaic model, we must understand that the detailed structure of the cell membrane was studied only after the invention of electron microscope. Until then, it started in the lab with biochemical analysis on the human red blood cells. Studies revealed it was mainly composed of lipids and proteins, and the lipids are arranged in a bilayer. We can see here the lipids are arranged in a bilayer. With further biochemical studies, it revealed that the major lipids in the bilayer are phospholipids. It forms the continuous structural architecture of the cell membrane. These phospholipids are amphipathic in nature. What does it mean? When chemical compounds possess both polar and non-polar regions, they are called amphipathic molecules. Now, phospholipids are amphipathic molecules because each molecule consists of a polar head and two non-polar tails within the same molecule. Polar head is called the hydrophilic head, that means water loving. Why? Because it contains an active phosphate group which is polar and attracted to water. In case of hydrophobic tails, phobia means fear and hydrophobia means fear of water. These are fatty acid chains that repel water. The amphipathic nature allows them to spontaneously form a bilayer in the aqueous environment with their heads facing the outside of the lipid bilayer and the tails towards the inner part. This ensures that the nonpolar tail of saturated hydrocarbons is protected from the aqueous environment. Now, the major phospholipids that are present in the cell membrane includes phosphatidylcholine, phosphatidylserine, and phosphatidylethanolamines. Another distinguished feature of the cell membrane is that it also contains cholesterol. Eukaryotic cell membranes, especially the animal cells, contain large amount of cholesterol. These cholesterol molecules are more rigid than the phospholipids. These cholesterol molecules act as fluidity buffer. It tucks itself between the tails of two phospholipid molecules, and this helps maintain the stability of the membrane structure. Biochemical studies revealed the presence of different type of proteins in the lipid bilayer. Now, depending on the ease of extraction, the membrane proteins can be classified as integral and peripheral proteins. The integral, also known as the intrinsic proteins, are tightly bound to the phospholipid bilayer. Now, here we can see this is an integral or intrinsic protein, and these are tightly bound to the phospholipid bilayer. Now, because they are tightly bound to the phospholipid bilayer, they cannot be easily removed from the membrane. Now, they are of two types. Some of them are partially buried and the others are completely buried or totally buried. Now, some integral proteins are completely buried across the thickness of the membrane, known as the transmembrane protein or tunnel protein or channel proteins. Now, this is one classic illustration of a transmembrane protein that is completely buried across the thickness of the membrane. The second type of the protein that are found on the cell membrane is the peripheral protein, also known as the extrinsic protein. They lie on the surface of the membrane. Now, the surface can be on the intracellular side of the membrane, or it can also be on the extracellular side of the lipid bilayer. Now, because they are loosely attached to on the surface of the lipid bilayer, they can be separated easily. One such example is spectrin. These are extrinsic proteins on the cytosolic surface of the red blood cells. So cytosolic surface means this surface. So these are the extrinsic proteins on the cytosolic surface of the red blood cells. They are part of cytoskeleton and they provide structural integrity and stability, and they also help maintaining the biconcave shape of the human red blood cell. But are these proteins and lipids present uniformly across the cellular membranes of all cell types? The answer is no. The ratio of the protein and the lipid varies considerably in different cell types. In your textbook, the exact percentage of proteins and lipids for the cell membrane of the human red blood cells are given, and that is approximately 52% protein and 40% lipids, the 8% being other elements such as carbohydrates. But we shall also look at the distribution of these things among the other cells to understand its significance. Here we can see that the lipids in this chart, the lipids are denoted by this orange part, and the protein components are denoted, the percentages are denoted with the blue color. Now here we can see that the myelin sheath, it contains considerably high amount of lipids compared to the proteins, whereas in case of inner mitochondrial membrane, the proteins are much higher than the lipids. From here we can actually conclude that if the primary job of the cell membrane is insulation, it contains higher lipid. For example, in myelin sheath we can see there are higher lipids because the primary job of the myelin sheath cell membrane is to provide the insulation. And if the cell membrane is occupied with extreme metabolic activity, for example, in case of inner mitochondrial membrane, which contains more proteins, because inner mitochondrial membranes is packed with enzymes for electron transport cell. So high activity means high protein, for example the inner mitochondrial membrane. Whereas for example in case of red blood cells, the cell membrane primarily provides stability and proper transport, so the lipid to protein ratio is nearly balanced, that is 52% to 40%. Alongside the major components like lipids and proteins, there is presence of carbohydrates. Oligosaccharides of the glycolipids and glycoproteins are present on the outer surface of the membrane. So these are glycolipids and these are glycoproteins. How can we say that? When the sugar chains are attached to the proteins, the membrane proteins, they are called the glycoproteins. And when the sugar chains are attached to the lipid molecules, they are called the glycolipids. Now, these oligosaccharides help in cell-to-cell recognition mechanism. One such example is during the fertilization, where sperm and the egg cells recognize each other through cell-to-cell recognition provided by the carbohydrate chains present on the glycolipids and glycoproteins. Now that we know that there were proteins and lipids in the bilayer, the question comes, how were they arranged? Were they a solid sandwich or something else? After a few models that were rejected by the scientist community, there was finally a model that explained the elasticity and the selective permeability of the plasma membrane. This is called the Fluid Mosaic Model. This model was proposed by S. J. Singer and Garth L. Nicholson in the year 1972. This is the latest and most widely accepted one. According to this model, the proteins are embedded as mosaic patterns in the phospholipid layer. These protein molecules, these are the protein molecules, these are embedded in the mosaic pattern in the phospholipid bilayer. Now imagine the cell membrane as a sea of oil where proteins are floating like icebergs, constantly shifting and moving. So this is called the protein icebergs in the sea of phospholipids. The protein icebergs in the sea of phospholipids. The membrane is not solid, it's quasi-fluid in nature, means semi-fluid or partly fluid. This fluid nature is necessarily important for cellular functioning such as cell growth, formation of intracellular junctions, secretion, endocytosis, cell divisions, etc. Phospholipid layer provides fluidity to cell membrane because the phospholipids are rich in unsaturated fatty acids. Now earlier we saw that the phospholipid molecules contain saturated fatty acid chains that is protected in the lipid bilayer from aqueous environment. In addition to these saturated fatty acid chains, it also contains a chain of unsaturated fatty acids. The presence of this cis double bond in this creates a physical bend or kink in the fatty acid tail. Now, this is a chemical composition of the phospholipid called phosphatidyl choline. Now, because of these kinks, the phospholipid molecules cannot line up perfectly side by side. You can see here when the phospholipid fatty acid tails are consisting of saturated fatty acids, they can be tightly packed. There is no gap between them. So these are densely packed. But when these are composed of unsaturated fatty acid tails, these kinks, we can see here, these kinks, they act as spacers. They push the neighboring cell or the neighboring phospholipid molecules away, preventing it from tight packaging. Now, this keeps the membrane in a liquid like state even at lower temperatures. The quasi-fluid nature of the lipid enables the lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. Now there are three types of movements that is generally present for proteins and lipids in the bilayer. These are lateral diffusion, rotational diffusion, and a very rare cases of flip-flop movement. Now let us understand each of these one by one. In case of lateral diffusion, the proteins and the lipids, these are phospholipid molecules arranged in a bilayer, and consider it as an integral protein buried in the phospholipid bilayer. So in case of lateral diffusion, these proteins and the lipid molecules, they move laterally along across the membrane. In case of rotational diffusion, suppose these lipid molecules, it rotates along its axis. So this lipid molecule, it rotates along its axis. Now, both these movements are rapid movements and are very frequent. The flip-flop movement is extremely rare, where one phospholipid molecule moves from one monolayer to the second monolayer, and this is energetically unfavorable. Now, the fluid mosaic model explains the asymmetrical structure of the lipid bilayer. Carbohydrates are present on the outer surface and spectrin on the inside. For the example, if we see this illustration, we can see that both sides of this lipid bilayer is not symmetrical. These are the oligosaccharides present on the extracellular surface, and there are peripheral proteins, let's say spectrin, on the intracellular surface. So there is no symmetry. Different things are present on different sides of the lipid bilayer. So it explains the asymmetrical structure of the lipid bilayer. The fluid mosaic models also explain the selective permeability of the cell membrane. It selects what passes through based on size and solubility. So basically they act as gatekeepers. Now the question is, how do different molecules actually cross the plasma membrane? So in the next session, we will move from structure to function. We will break down the active and passive transport and other forms of transport. So till then, study hard and take care. Thank you.